The floor is yours, sir.
Oh, thank you so much. So I start with a small, like short presentation, like 15 minutes. So
yes, at the 15-minute mark or 20-minute mark, we will.
Okay. You will stop me.
Yes, because we wanna be mindful of time and also having people.
Usually, I put an autopilot so for these presentations and stuff. So let's go autopilot. Okay, so it's a great pleasure to be here. First of all, thank you so much for your invitation. So, okay, I'm not going to really talk about details and technical things here, but just give you a little bit of overview of what we have done before and what we are doing right now, and what is the notion of biofabrication or tissue engineering and organ on a chip. So as you see on the screen, I'm assistant professor at the University of Montreal in the Department of Pharmacology and Physiology and Institute of Biomedical Engineering. My lab is located in the hospital, in the Moms and Children Hospital of Montreal, which we call it Sainte-Justine. So I think it's pretty much all the cities, they have this kind of hospitals for kids and moms. So, and also I'm holding a chair position in one of the, like these innovation incubators, like centers in Montreal called TransMedTech. So my chair position is in 3D bioprinting and regenerative medicine. So I started my lab just before the pandemic. My background is, I was not really lucky, but I mean, c'est la vie en français, in French. So life goes on. So yeah, basically my background is in chemical engineering. So I'm a chemical engineering training. I did my bachelor like long ago, as Amir mentioned. You're very old, like, because he didn't see me for 30 years now. So, and then I did a PhD also in Iran in biomedical engineering. I moved to Canada in 29. I went to University of Ottawa. I did a research associate position there for a year and a half, for almost a year. Working more on fabrication of membranes for water purification and gas sweetening and this kind of, because I was chemical engineer, right? So and then I moved to University of Montreal. I did my second PhD there in biomedical engineering. From what I learned, from my master's, my prior experience, I started, switching to biomedical engineering, and then I did my postdoc at University of Toronto, and then I moved back to Montreal when I got this position as assistant professor at the University of Montreal. Okay, so I start my, let me just put my timer on. Okay. Yes. So I always start my talk with this, the story of Luke. Luke, at the age of 10, he was really like sick, and he was actually like suffering from a neurodegenerative disease that led to malfunctioning of his bladder and his kidneys. So he couldn't enjoy real life with his peers, and he had to go undergone too many surgeries during his time, his teenager time. So thanks to tissue engineering, this is from BBC News in 2018, he got a new bladder from his own cells, made up of his own cells, and now at the age of, I think this is 2018, now 31, but now he's enjoying his life. He has a healthy life. He is a wrestling coach. He continued his studies in the school. And so this is the promise of tissue engineering, basically. So what we really do, we help people to kind of, to decrease their suffer or their health problems. So basically, if we want to talk about tissue failure, I mean, tissue failure is a really tremendous clinical burden worldwide. So as you can see, even like, alone in US, each year there are 5 million surgeries to replace damaged organs and tissues. And this accounts for many different tissues like bones, arteries, veins, internal organs like kidney, and neurons or other tissues in the body. And as you can see in this graph on the right-hand side, the supply is always higher than the donor. So there is a big waiting list for receiving, transplant or tissue replacement. So there is one option to get the implants, artificial implants that are made up of metals or polymers or plastics or whatever. But the problem with the synthetic implants is that that they fail because of wear or fatigue or biocompatibility issue with the host organ. So for us as bioengineer or tissue engineer, so we actually we are dedicated to find a solution through bioengineering to create or build these artificial organs for replacement, for implantation in the body. So just I gave you one of our promises in tissue engineering is to make the artificial organs for transplantation. So this is really a long-term goal for us. So it may take another 30 to 50 years to have these tissues ready in the market for the surgeons to transplant them in the body. But we actually, as tissue engineer, we also work on these artificial tissue as a platform technology for drug discovery and disease modeling applications. So in case, I mean, we all know about the COVID issue during the past 2 years. So, but that was really accelerated, accelerated pipeline for the vaccine. But usually, if any medication, any medications comes from the lab, it takes around 10 to 15 years to be commercialized or to become marketed, right? So, and this pipeline, categorized through three sections, three different pathways. The first one is just evaluating the different medications in the lab, that we call it in vitro evaluation. And then in vitro evaluation, you have many candidates, right? And then once you screen your different options and candidates, you actually screen them down to a couple of candidates, or maybe 10 or 20 candidates, and then you do some studies in animals, right? What we call it in vivo study. And then after that, you go through different, phases of clinical application in the small population of human. We call it clinical phase 1 and phase 2, a little bit larger population, and then we go phase 3. And then after that, when we make sure that this drug or these drug candidates are efficient and safe, so then we file it for the FDA approval. FDA, for those who for those who don't know about, is Food and Drug Administration, but it could be in Canada, Health Canada, or in Europe, European kind of regulatory agency to validate our study. So as you can see in this kind of graph, images, pharmaceutical company, they put billions of dollars each year for R&D to bring new drugs to the market. But most of these drugs, unfortunately, they fail. As you see in this graph, they fail because of efficacy and safety issues. So one of the famous examples of these drug kind of withdrawal from the market is Vioxx, that was meant to heal the bone problem or arthritis or kind of, for arthritis, but really the side effect was cardiac toxicity or infarction or heart attack. So many people, like more than 20,000 people in US only, they lost their life because of this side effect. And Merck, the company, the Merck company that marketed this medication, really, forced to be compensate these families for losing their loved ones because of the side effect of these medications. So why this happened? During this pipeline that I show you on the top of this slide, the pharmaceutical companies, or even in the laboratory, we use basically cells in the Petri dish, to test the drugs. Basically, it's a monolayer 2D cell culture, we call it. Or in parallel, as I mentioned, we tested after that in the animal models. But both of these models cannot really mimic what we have in our body, right? Our body or our tissue is 3D, is not 2D. And we are different from rats or any other animals, like pigs or sheep or cats or whatever animal models that we're using. So this is really, we lack reliable models to test the drugs. So by that, as bioengineer or tissue engineer, we use these artificial organs that we actually build in the lab as a platform technology that we call it organ on a chip, small mini tissues on chip to test the drug. So there are some pros and cons for each of these platforms. For animal models, you can see that I listed here, but for organ on a chip, these platforms are really reductionist models. So they are inexpensive. So we can use the human cells. We can use, for example, we can do personalized medicine. For example, if a person has a rare disease or has a specific disease, we can get his own cells and then model his disease on chip. And then we test different drugs, and then even we can propose which dose he can take. So this is personalized medicine or precision medicine. These platforms are high throughput, so you can do many tests at a time or many drugs at a time, and then screen the bad ones and take the good one to the next step. So there are lots of pros for this kind of platform. Or even you can connect these tissues on chip together, so you can have human body on a chip or human on a chip. This was first shown in Harvard University, that these individual tissues can be connected to each other. And then we can see the effect of different, side effects. For example, if a medication is meant to heal, for example, skin, or heal, for example, bone, it doesn't have any side effect on heart or on liver, right? So these tissues, even we can see the downstream and upstream kind of effects on human on a chip. So my lab, what we do in the lab, we work around these areas that I mentioned. So we make artificial tissues. So we use materials, we synthesize new materials, we actually use human cells, and then we mix these together. We have a cocktail, and then we use biofabrication techniques. The newest one, the recent one, is bioprinting, for example, to print different structures, to print heart valves, to print artery, to print cardiac patch, or different tissues, right? Kidney, or microfluidic. We use microfluidic to make this, to kind of put these organs on chip that are exposed to the fluid, so we can mimic what is going on in the body. So we use these two kind of. So I just passed this. I just want to give you one example. For example, here in one of our projects, we try to mimic the, let's get back, to actually mimic the artery, right? Using electrospinning technique, which is one of the fabrication techniques. So artery has 3 layers. So we try to mimic this structure of the artery and come up with the small diameter vascular graft. We call it vascular prosthesis. So what we did is that we designed these different layers, and we optimized the processing condition of this fabrication technique that is called electrospinning to get this fibrous structure. Why fibers? Because our body, all of our tissues are composed of fibers. If we look at our tissues under the microscope, we see fibers. So that's why we create these fibers to exactly mimic what we see in our tissues. So we have these different fibers. I don't want to go to the details, but we come up at the end. It's at this picture on the bottom left. It's a small diameter vascular graft that we actually make it, and then we plant it in the animal model, in the sheep. And unfortunately, it failed. It didn't work. So we have to report also the negative results. So the problem is that the mechanical property of these grafts that they made was not really close to the native artery because of the mechanical mismatch between these material that we use and the native artery. So it failed. So at the end of the day, after long-term evaluation, it was blocked and the sheep died, right? So this was one of the projects that I wanted to see. So another project, I talked about fibers. So we are really interested to make fibers with different techniques. With my friend Mohsen Akbari at University of Victoria, we made a new technique called Microfluidic Bioprinting. Basically, he developed this technology, and then we use this to have these different fibers with different shapes. For example, solid fibers, hollow fibers with different cross sections, with multi-materials, as you can see these SEM images here. And also we can braid them, we can weave them, we can make different structures. For example, if we weave them, we can have skin patch or cardiac patch, or to heal the heart, or to heal the wound, for example, for skin. So these are the fibers that you can see. We can bioprint or fabricate different materials, 2 different materials. As I said, you can have skin patch, and you can, as you can see in the SEM here, these fibers can be braided or can be woven. And then we have the cells, different cell type. We mix them with this hydrogel, so we can incorporate cells with these fibers, and then we can start building the tissues in 3D. These are the cells, for example, myoblast, and on the bottom cardiac blasts, cardiomyocyte that create the heart tissue, for example, with these fibers, right? Let me know how much time I have. I mean,
you're almost done.
I have my time here. I started 19. Okay, so 15 minutes. Okay, so let's. I pass because I'm just speaking very slowly. Anyway, so this project was really cool. During my postdoc, I did this. So as you can see, this is the one of the chambers of the heart. This is the left side of the heart that really is responsible to pump the blood out of to the body, right? We call it left ventricle, Battenet chap, left ventricle, right? So we actually look at the left ventricle. So it really is composed of different fibers from the bottom of the heart to the top of the heart. So we try to mimic this structure in the lab. So we use fabrication technique called soft lithography. And then we created these channels that really mimics these patterns, you know, these parallel fibers on the heart. And then we put the heart cells, heart muscle cells that we call it cardiomyocyte inside these channels. And then once you have these scaffold, we roll it around a mandrel, like a stick, like a small stick, like a mandrel. So we have this mini heart. And then we use this mini heart to test the drug. We connected this mini heart to a perfusion system. So the flow can go through this mini heart. And then what we show here is that this heart can respond to, first of all, can beat and can also pump a little bit of the fluid, the media that we put there out of this left ventricle. So this is a platform that we can use, test the drugs now on it. Instead of testing the drug on the animals, we can test the drugs on this heart that we use the stem cells. We use the cardiomyocyte that we got it from the stem cells. It's a human cell, so it's really reliable platform. So I just want to cut down my presentation, go to the recent work that we did. So basically, I just want to summarize. We make different structures, fibers using 3D bioprinting techniques. So to make these 3D structures or tissues or organs, we create different biofabrication technique like microfluidic printing. Like recently, I use sound-induced printing. Using sound, we can pattern cells, we can patterns, we can have different tissues, and then extrusion-based printing. And then we can make cardiac patch, or as I mentioned, like a left ventricle for drug screening or testing. One of my students recently started to create heart valves by using bioprinting technique. Maybe this is interesting for you, how we print, for example, a tissue. So basically what he did, he actually created or synthesized or made a new material that we call it bio ink. And then he started printing this bio ink in the shape of a heart valve. So now he's working for evaluating this in vitro using different assays. And then later on at the end of his project, as you can see in this schematic, is going to test in animal, and also is going to test it as a platform technology for drug screening and disease modeling. So this is a new project that we started. This is a sound-induced printing technology that we can pattern tissues. I'm gonna stop in 1 minute. So as you can see, using sound waves, you can pattern cells on the left-hand side. And this is, I put the extrusion-based printing on the right-hand side to compare, how we compare different techniques. The extrusion base on the right, the sound induced printing is on the left hand side. So we started, patterning cells and hydrogels to get different tissues. So this is really a cool technology that we are working on. So as you can see on the left-hand side, the video is still continuing. Okay, so I pass. And also I have a little bit of hat of entrepreneurship. So one of the technologies that we have in the lab is creating a 3D in vitro model, as I mentioned, a 3D tissue, a small tissue for drug screening, but this time for oncology purposes, for drugs that we actually need for treating the cancer patients. So we have this, we are working on this startup, we call we chose a name Beehive Biosciences, for making in vitro cell culture platform that are more representative of the living tissues. So it's still in earlier stages, but we are trying to get some more funding to really recruit some people and bring it to the next stage. So that's it for me for the part of presentation that I had. So my lab is funded from different, entities, funding agencies in Canada, NSERC, TransMedTech, ThéCell, and also Quebec government, and I have lots of collaborators. The main one is Mohsen Akbari. Maybe is a good candidate for your next tech talk at University of Victoria. He has more stories than me to share with you. Anyway, thank you so much. I'm happy to take your questions. Amir, you're muted.
Yes, this time it was my turn.
After 2 years of Zoom.
Yes. Yes, so you mentioned Mohsen a few times, Mohsen Akbari. Yeah, I was at UVic and I know Mohsen, the other Mohsen, through that, for UVic. So it's good to shout out to Mohsen Akbari as well.
Exactly. A good friend of mine.
Yes, he's. I remember when he first came to UVic for his PhD studies. Yeah, definitely an option to consider for. So Mohsen Vafadous will definitely reach out to him. So before we get to photos, we have a tradition of taking a group photo. I said before going to photos, before going to questions. So turn on your cameras if you want to be in the group photo.
We see you as a focus screen, on a focus screen, right?
What do you mean?
I mean, I don't see everyone. Let me just see. Oh, okay, yeah, now it's
awesome. All right, everyone ready? 1, 2, and 3. Okay. Oh, by the way, we have Amir Nouri joining us as well. So Amin Nouri, sorry. Would you like to introduce yourself? Tell us what you do.
Hi, my name is Amin Nouri. I met Dr. Savoji in 2006 in Sheriff University. So it's
really?
You were my thermodynamics TA. So
my goodness! Oh,
so
wow!
It's great to see you. I'm in hydrogen fuel cells now, far from your field, but great to see you and everyone. And thank you for your talk.
Where are you, Amir, now? Sorry.
Burnaby, Vancouver, pretty much.
Oh, okay. So you are on the West Coast. Okay.
Yeah.
Wow. I already forgot all the thermodynamics formula and equations. You have now to kind of, remind me what was the van der Waals equation or this kind of things. I'm far now from those chemical engineering core courses.
Yeah, that's quite a few years. Surush also joined us. Surush, do you want to introduce yourself too? Surush, yeah.
Hello.
Hi.
Yes, I'm Surush Rasti. I thought the meeting is over. I thought.
Nope, we're still here.
I live in Waterloo. I have a PhD in electrical engineering. I got it 2 years ago. And now I'm doing 5G, mostly RFIC or circuit design in the industry.
Awesome. Thanks for coming.
Thanks.
All right. Time for questions, comments, anything that you have in mind. Go for it. We have a very quiet crowd. It usually takes a little bit of effort to break the ice here. So I'm gonna.
I have one question, if I
Sure.
may. So you mentioned this human on the chip idea or technology. So is the ultimate goal to replace animal testing, or will it be an idea to pre-screen a bunch of stuff and then go to animal testing? Like, what's your vision on that?
I mean, very great question. Thank you so much. So we don't claim to replace animals, but we claim that we reduce animal consumption first in terms of animal welfare. And second of all, we say that we use them in parallel, as you mentioned, So we get the results, we get the performance and safety and efficacy of these drugs on the chip, and then we test them on the animal. By using these chips, we can, first of all, as I said, decrease the number of animals that we use. And second of all, we are much closer to the human genetics, pathology, and what we really have in human tissues, right? So then we can validate. Even we can validate. Animal models are not good for validation, even on the chip results. We can validate with the clinical results. If there is, for example, a known medication or drugs already in the market, So we can do that. But for unknown medications or candidates, drug candidates, we can do animal testing in parallel. So that's why those people who work in biology, I think we have one of CPD. They don't believe us. When we say human on a chip or in vitro models or this kind of things. They don't believe us. The people who works with animal, they really don't buy this kind of stories that we say. I mean, they want to cut our kind of, salaries and monies. So come on, we are not competing, Safide. You do your job. You do animal study. You do on a chip study.
Thank you. Actually, it was really nice presentation. I really love that. It was the first time that I could see it from beginning, because I always miss your presentation. I signed up for, but I never completed them. So it was amazing. Thank you. I don't disagree at all. Actually, it was quite nice. We also want to reduce the number of animals we use in the lab. So we have another model of for drug screening and also for metastasis model, which is quite interesting. I don't know if you have ever heard of chicken embryo model.
Yes.
So we grow chicken embryos and we put on plants, different type of cells, engineered cells, control cells, let them grow. Sometimes we inject, so we monitor cell migration to the brain and other organs. But still, even though it's very close to a real animal, it's very complicated story for cancer research. Because that the cell is in communication with neighbor cells. Exactly. And so it's for us, actually, I think it's quite difficult to mimic that to a true animal. However, we do our best to get the closest.
Exactly. So just follow up what you said, on a chip, also the same problem, you have only one cell type or maximum 2 cell type, co-culture them together, the immune system is not there, the neural central nervous system is not there. Many things are missing, right? So, and this is something that always reviewers, when we write the grant and submit, they ask. Myself, my grants always are rejected because of one of these issues. Why you don't have complex, kind of, model. So, but the answer, because I got the advice from many good people, my mentors, Ali Khadim Hosseini, Milica Radizik, all of these guys are very, 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 pioneer in this field. So we don't really use complex, we actually designed the system to answer the question. So it should be relevant to the question that we are asking. If the question that we are asking is the interaction between, for example, a medication and cardiomyocyte or cancer cell, so we do that. We do only one cell type. But if your question is the interaction with the immune system, and then, for example, cancer cells and this, then we add the complexity to the system. So from the beginning, we don't need to do that. We don't need that complexity. We have to start, step by step. But I was wondering, if these chicken embryos, you grow them in the lab, and then in the weekend you forget to come back, they become chicken, and then what?
No, well, no, actually, no, they don't. So we have a limitation. In order to keep the whole work ethical, we are not allowed to keep them alive more than 21 days. So when we purchase the eggs, we grow them, we actually take care of them for a week or so. So we start injecting them, we leave them for like maybe another week or so. And then we will have a very elaborate and complete organ that has grown, a real tumor, a 3D tumor. And then my today work was completely just dissecting chickens, embryos. So maybe hundreds of them to get enough organs to study any migration or metastasis of primary cells to brain, to lung, and to liver and bone as well. So then when we dissect them and finish them off in the day 21, still not a full animal, it's still ethical. And we still got our migration studies and metastasis studies done. It's quite inexpensive way of doing animal studies, and very short run, less than a month, you get good number of animals or embryos.
And in terms of epigenetic, are they close to human or?
Well, it's like a xenograft thing, so it's not meant to be completely similar. We just use them as a matrix, as an environment to grow our cells in.
Perfect.
And as long as they have vasculature, sorry, I cut you, but they have vasculature, and it helps us. So that's quite nice.
That's what we do. We try to build in vasculature inside because if we make the thick tissue, then you have necrosis, hypoxia, and this kind of, because, sorry, I use terminology. Some people are coming from computer science. So when you have a thick tissue, inside the oxygen nutrients cannot reach really to the core of your tissue, right? So then the cell die. So that's why we try to build vasculature inside and to bring this oxygen and nutrients inside this tissue to keep this tissue for a longer term in the lab, right? But for chicken embryo, khoda dadiye dige,
it's a natural
vasculature is there.
Yes. Yes.
Yeah.
Awesome. All right. I enjoyed this conversation. Who else wants to go next?
Do not hesitate. Don't ask technical question. Ask as you wish. Any questions.
Definitely.
I can ask a follow-up question. Has there been, what's a success story in human on the chip? Has it actually replaced any animal testing so far, or is it a vision that it will do it in the future?
No, very good questions. I think you read that. Probably you read some of the stories because you have some kind of foundation background about this. Your questions are to the point. Yeah, no. So the first time this was done in Wyss Institute in Harvard. So they try to connect different tissues together, and then they added also biosensor to detect the biomarkers, oxygen level, pH levels, and everything in different tissues, for example, liver or heart, or because, you know, so each tissue has their own function and biomarkers, so you need to also integrate the biosensors, right? So and also it's very complicated because you have pumps, you have you know, these sensors, everything. So anyway, so they tried to do that, and it was successful because in terms of you know, just simply connecting these tissues and see the effect of, for example, upstream kind of, you know, if they administer, for example, a drug on the upstream tissue, on the downstream tissue, what will happen? Or for example, for cancer extravasation, for example, migration of the cancer cells from tumor to another tissue and this kind of things. They did lots of these studies as well. So in terms of replicating, because your question was specifically on how reliable are these human on a chip for like or, you know, like similarity to animal testing. To be honest, it is not really. It's not really, because it, you know, it's simplified model, right? It's a still simplified model. We don't have immune system. We don't have, as I said, neural central nervous system. We don't have many things are missing. But you know, there is a hope. There is a vision, as you mentioned, that we can have this complexity in the future. But there are many startups. For example, from a lab in MIT, Linda Griffith is a pioneer in multi-organ on a chip platform. The company is CN Bio, that they have these different, you know, like now they go up to 10 organs or on chip, right? So you can still, like, people trying to first of all increase the number of the organs that they can connect together, and increase the number of the cells that they can co-culture together on each well or on each platform. For example, you know, talking about heart contain like at least 4 or 5 different cells, but we always study cardiomyocyte. Maximum we add a little bit cardiac fibroblasts. But there is a smooth muscle cells there, there's endothelial cells, there are other immune cells, lots of pericytes, lots of cells are in heart tissue. But maximum we have 2 cell type, cardiomyocyte, cardiac fibroblasts, right? So yes, now this is the vision, but people trying to add more complexity to this system in terms of number of the organs that are connected, and in terms of number of the cells on each organ.
Thank you so much. Appreciate it.
Thank you.
Awesome. Anaita.
Yeah, thank you for the presentation. I really enjoyed it. And though I do not have any background in biology, just the general knowledge, it was very fascinating.
Me too, I don't have. I'm an engineer.
Yeah, but after these many years of work,
I may pass your question if it's about biology to Sapid.
No, it's not. It's just I'm saying that if my question is like does not is not reasonable, maybe like it's because I do not have the background. So like you mentioned for human on chip that there are just limited organs and like no central neural system. So I wonder if any group has ever tried tissues that are similar to the brain tissues. I mean, like the cells that are similar to brain cells. Because like you mentioned other organs, but I didn't.
We do have brain on a chip. I mean, some people try to mimic brain on a chip, and they try to also, there are lots of works on blood-brain barrier on chip. Because the important thing, because in brain you have this kind of membrane or barrier that the medications or drug goes through, and then they reach the target, which are like nerve cells or brain cells or something. So people trying to mimic exactly or really model this membrane that they call it blood-brain barrier, and there are many different platforms, but all of them they have this membrane, because basically blood-brain barrier is composed of this membrane. Yes, brain on a chip we have, and neuron on a chip we have. We have blood-brain barrier also, and we have different kind of brain cancers, glioblastoma, and this kind of things on chip as well. So there are, whatever you think people are doing, So even when we write a grant, it's really difficult to come up with a new idea. Somebody is doing this in the world.
Like my next question is that then, okay, right now these human on chip systems are like very simple, but I wonder in future when if they get more complex or whatever, what's the limit for the brain on the chip because of the ethics? Right now we do not exactly know what's how conscious is related to brain, and those kind of things. How much of the brain are we allowed to implement on a chip so that we cannot call it human conscious, or so that we can be sure that we are not torturing a creature?
In terms of ethical issues, since we are using the stem cell derived, we actually get the stem cells, that these cells comes from skins, from biofluid like saliva, blood, or any other biopsy. If somebody doing surgery or something, we get the cells from any tissues, not brain. And then we actually make them brain cells, make them heart cells, make them any cells that you can think of, right? So in terms of ethical issue, and now with the induced pluripotent stem cells, the ethical issue of the embryonic stem cells is already solved, but the induced pluripotent stem cells also has still some problem, like a tumor formation and this kind of things. But the ethical issue is kind of not a problem right now, because we get the cells from saliva, blood, skin, whatever, and then we make them whatever cells that we want, and like brain. But if I understood your question correctly, this is the ethical issue that we actually faced before, but now we don't have with the stem cells, using the stem cells.
Actually, what I meant was that one day we may have brain on the chip, the whole brain, or most parts of the brain. So in that case, although they are produced from stem cells, they will make a brain, a full brain. And then we can say that human is much about brain, right? Like artificial intelligence. So at that stage, if we ever are planning to get to that stage, or we will get to that stage, then in that stage, will it be ethical to run experiments on that brain, because we can consider it as kind of like a human being? It's very it's not reality, but I'm just thinking maybe too much.
I understand. You're talking about, for example, I don't think that would be an issue, because these are all experimental models. There is no soul, as you're talking about extra kind of
consciousness
and
self-awareness.
Yes. No, these are all experimental models, and we don't get to have really like a living human brain on a chip. We have living human brain, but these are experimental model. There's no ethical issue in that sense, I'm just making a joke. We can have zombies on chip one day. But no, I mean, there is no problem with the ethical issues, you know, in that sense. I understand your point. You say that there might be, because we have human brain on a chip, but then the question comes, when I had this physiology course during my master in Iran at Sharif. So there was a professor from University of Tehran who taught us physiology, and he was kind of debating which one is important, heart or brain. Then I may ask, or that professor may ask you this question: heart is important, heart is ruling the brain, or brain is ruling the heart? Which one is important, more important? So he was, I mean, this goes back to 20 years ago when I was doing my master's, but this is important to know. I don't know the answer. Who knows the answer? Brain is ruling the heart, or heart?
So this can be a very philosophical question too.
Exactly.
Because, as in our literature, it's taught as the battle between heart and mind, جدال عقل و عشق, love and mind.
Exactly.
And stuff.
Exactly. But for example, if somebody died, I mean, if the brain, if somebody has stroke, as long as the heart beats, they keep them in the ICU, right? Or if somebody goes, so it seems that when the heart is still beating, you know, there is an ethical issue of kind of, you know, how do you say, you know, take the patient from the machine, right? So I don't know. This is to be debated. Please go ahead.
Well, we are gonna keep that debate going, but before we go to Mohammed, there's one thing
Yeah.
to mention is that at this time we ask our guests to think about what our takeaway should be from this meeting. We're going to get back to you at the end for that. And we also have Nilufar joining us. Nilufar, can you introduce yourself? Nilufar.
Hi, can you hear me?
Yes.
Okay, hi. It's my pleasure that I'm among you. And, you know, yes, as you know, my name is Nilufar, and I've graduated from Materials Engineering, and I'm from Iran now.
Awesome. Are you in Vancouver now, or are you somewhere else?
No, no, I'm in Iran right now.
Oh, nice.
Yep.
Well, thank you.
Thanks for joining us. What time is it? 6 o'clock?
Yep. Yes, 6 o'clock.
Wow. I really applaud your dedication. Thanks for joining us.
Thanks you.
And with that, we go to Mohammed.
Yeah, thank you. I apologize me for at the first of meeting I couldn't open the microphone, but this for this question, the neuroscientist believes that all things in the brain. Heart is nothing. Heart is just beating and pumping the blood for all body, but all feeling is in the brain. Yeah, heart is just the symbol of the feeling, but is not the actual feeling in the heart. All thing is in the brain.
Awesome.
So then the question that Amir mentioned, you know, what is love? Who decides to fall in love, brain or heart? Or this is very philosophical. Cut this from the video.
Actually, I'll keep it. From a technical discussion, that's why we call it Tech Talk with a K, not TECH, because sometimes we go to other places too.
I think that's why people like my post, because they thought it's TED Talk.
Well, that's
So the Houman Savoji is going to give a TED talk. It's very important.
that. But yeah, we had one of our guests. He spoke at TED Talk before he graduated to speak at Tech Talk. So we kind of take pride on that he had to pass that to come here.
Yeah, when Mohsen invited me, I thought it's TED Talk. I was so excited, and then I was, preparing my pitch, and then.
Well, it's more important now. You can tell.
But this is much better than TED Talk. I mean, it's friendly. Right? Yeah, much better. I really enjoy it.
Thank you. All right, folks, we still have 20 more minutes to go, so jump in.
So just to complete what Mohammed mentioned about neuroscientists, like for artificial intelligence, computer science is also the same. I mean, like we assume that most of the things with human is the brain. That's why artificial intelligence, they try to mimic the brain, not like heart or other parts. And this consciousness is also a ethical question and like problem in the field of artificial intelligence. Not now, because now we are not there yet. It's fully like a brain, but for future it is. And like lots of sci-fi movies and things have been made on that.
Yeah, Yuval, not Yuval. What was the name? Harari. Harari also is really scared of AI in the future, like the ethical issue around.
Yes.
Yeah. Not what was his name? First name?
Yuval.
Yeah,
not Yuval.
Yeah, exactly. I was a bit, but I actually watched many of his videos on YouTube. So he's really like concerned about this.
Awesome. All right, I'm gonna pick on someone to jump in since people are kind of quiet. So I'm gonna pick on my easiest target, Mohsen. If he's close.
I think there is a problem.
There's an issue if your mic, we can't hear you. Very far away.
Yeah.
As if you're at the end of you're in the next room speaking. It's not like you're in this room. No.
There's a technical problem with your sound.
Maybe just use the computer microphone as opposed to whatever you're using now.
No.
Nope. Yeah, technology sometimes fails us, Mohsen.
Then we are talking about AI.
Yeah.
How about now?
Yes.
Perfect.
Much better.
So my question is, how long do you think it would take to this technology take place of the animal killings or animal testing?
Sacrification. Yeah, so yeah, that's a good question. I don't think it's a short-term kind of short-term thing to happen, because we still need animals to test and to validate some of our kind of finding on these on a chip platforms. But the aim, as I mentioned, is just to reduce animal consumption and also get closer to what we have in pathology and in terms of genetics and pathology of the human body on a chip. But I think it's a long-term kind of vision or goal for us to really completely replace animal models. So for me, as somebody who's working on this in this field, I can say maybe in 20 or 30 years from now, we can do such a thing. Once we have a complete human on a chip with all the features that we mentioned, you know, immune systems, nervous systems, all these vascular networks, all these tissues can be developed to the point that they are mature and functional, right? Even heart, when we do it on a chip, in the beginning, it's not functional, it's not mature. So we have to mature the heart cells, because I'm talking more about heart because my focus is heart. So the heart cells that we get in the lab from stem cells, they are not mature. We have to stimulate them electrically, mechanically, chemically, or whatever to mature them. And then, so this is the case for other tissues as well, for neural tissues, for kidney, or for any other tissues that we're talking about. So the maturity of the cells and the functional properties of these tissues is a problem still. And also, as I mentioned, you know, the complexity is not still there. So we need to wait another maybe 20 years.
Right.
But you know, we actually made a business for ourselves, because the implantation takes 50 years. So we said that, okay, we are making this tissue, let's just use it, you know, make a business for ourselves.
Right, makes sense.
So can I follow up on that question? You're mentioning this this vision of 20 to 30 years timeline for it to come to fruition. How does it fare with making a startup out of it, making an entrepreneurship idea? Like who pays for what kind of investor pays for something that you say may take 30 years to come to fruition? How did you achieve that?
Yeah, a very good question. I mean, you know, the platform is much better than the 2D culture system. Now the pharmaceutical company, they use 2D culture system. So the pharmatech, they pay. Now Pfizer, Johnson & Johnson, I don't know, other Merck or other companies, they buy these technologies from a startup. And some of them, they acquire this startup, you know, to kind of, you know, Johnson & Johnson does this, for example, J-Lab, that kind of, you know, catalyzing the innovations and the stuff. They acquire this kind of a startup, and then they kind of, you know, bring it under the umbrella of the Johnson & Johnson, and then they use this platform. It's much better than the 2D culture still. It's not yet there as kind of, you know, full model, like mimicking what we have in the human body, but still it's kind of, you know, intermediate, you know, like between the 2D culture animal model, and can be used, you know, for some validation study and some screening, at least in the beginning of the pipeline, right? Pipeline, I mean the pipeline that I showed, you know, during that 3 to 5 years that they screen different, you know, candidates. So yes, the revenue is there. And it's a very, very hot area. It's booming actually. Like I have a review paper. I mean, not my own review, but my ex kind of lab member wrote a review paper on different, you know, startup companies at that time, like 2018. So he actually summarized all the startup companies in one review paper that they were working on that time on this technology. But now I can say that the number increased by 10 to 20 fold, you know, like the number that he reported. There were few of them in that review paper, several of them that he reported. And since now the ecosystem, like in all my internet connection is unstable. Can you hear me?
Yeah.
I got this notification. Anyway, so the ecosystem, the entrepreneurship, you know, they are in any country now, they're trying to support this kind of, you know, innovation and entrepreneurship aspect. So people, and in the beginning, government I mean, we use the non-dilutive funds and this kind of things. And later on, the investors comes, and most of them, you know, are pharmatech. You know, VCs from the pharmatech that they come and then they pay for these technologies if they really see the benefit. For example, one of the most important companies in the field is the Emulate. Emulate is a company spinned off from the lab in Harvard that first published on this kind of technology in 2010. The technology was introduced first, the terminology, organ on a chip, was introduced in a science paper from Harvard University in 2010, and then the company was a spin-off on long on a chip, because the paper was on long on a chip, the paper that was published in Science, and now the same company, Emulate, they have different platforms: liver on a chip, kidney on a chip, lung on a chip. And then they were the first company that FDA and also NIH, you know, started to really support them. And now the platform, the chip that they make, it's $5, but they sell it $500. Look at the value proposed. I mean, like the revenue that they're making. You know? So the chip basically is out of PDMS. It's a silicon. And the technique is fabrication technique. It's just soft lithography or any other fabrication technique. So material-wise and human resource-wise, you know, if we can just do the math, it's just less than $5. But then, you know, they sell it $500.
Wow. That's amazing. I hope that happens to your business as well.
No, we are, I mean, we are a bit late. Always we are. You have to be on the, you know, like forefront, you know, like pioneer. But now, no, I'm not, I'm a bit joking, but still, like there are rooms, you know, to develop new platforms, to optimize because not all of these platforms are kind of, you know, complete or, you know, answer all the questions. So, for example, we find a niche or find a gap in the field of, you know, in the mimicking the tumor environment. So that's why we have to come up with this idea, but I cannot disclose it now. But anyway, so hopefully, I mean, next TED Talk that Mohsen invite me, I will come and talk about our product.
Best of luck. Thank you.
Thank you.
Awesome. Soroush.
Hello. I have a question or two questions from Houman. The first question, so basically I'm a chip designer on electronic circuits, but when you say chip, so a chip to me is a silicon platform that's a planar thing and we grow something on it. But what do you mean by brain cell on chip? Is it a planar technology?
Planar means flat.
2D, yeah.
No, it's not 2D. That's a whole story that I'm just trying to say.
I missed
You know,
the first part of the talk.
Yeah, no problem. I mean, yes. So we have this chip. First of all, because you're working on microfab and stuff, so you have your silicon wafer. So we make the master mold from the. Sorry, I'm going to terminology with Soroush because he's working in this field. So we make the master mold out of the silicon wafer, and then we pour the PDMS on top of this master mold, for example, positive or negative mold, and then we have these channels or chambers on the PDMS, and then we make our platform. We kind of, you know, we can have two parts and then we bind them with after plasma treatment together. And so then you have a channel or a chamber or a well, whatever, sandwich between these two PDMS, PDMS. Then inside you can encapsulate your cells inside the hydrogel, right? So then it's 3D, it's not 2D. It's not on the surface of PDMS, it's
So you have
inside
a
this
channel,
chamber.
basically.
Yes, I have a channel that they're filled with hydrogels, and it's 3D. So the channel has widths and the height. And
But it's still the thickness is a few micron, right?
exactly. So that's something that our company comes kind of to address, because sometimes, not most of the time, as you mentioned, the thickness is micron size. It's not physiologically relevant, or it's not really close to what the tissue thickness is, right? So
How big is the
it's,
tissue thickness?
imagine the heart, for example, wall of the heart is let me just centimeter size, right? Or a tumor, look a tumor like grows, metastasize and the stuff. And Sepide was here, like we have a big tumor kind of, when we work on embryo. Sepide left? No, she's here. So anyway, so the niche is now here to actually mimic the size of the tissue that we have in the body. But it's not always the case. You can get the same, you can address your question on the mini tissues also, the micron size tissues. It depends on what question we're asking, So for example, in terms of cancer, we cannot really get, we believe, with those small cancer on a chip models that is micron size. So that's why we try to scale up. But in heart or other tissues, we may get the answers to our questions with the micron size tissues. It depends on the question that you're asking. But yes, to give you a sense, it's just a chip in the size of a loonie, and connected. It can be pumpless. It can be connected to the pump. You have the perfusion inside the channels. You can have fluid going on, or you can.
Is the same as microfluidics?
Yeah, it's microfluidic
It's just a different name
combined with stem cells, hydrogels, and tissue engineering. When the microfluidic is combined with the stem cell technology and hydrogels and this kind of, we call it organometry.
And my second question, thank you, was that you said there is no soul so I guess Seppi the question or Anahita.
No, cognition. If I talk about
does it mean that you don't even believe in your work? Because you
no, I was joking.
Imagine one day really you build a complete human. What would happen?
Wow, this is very ambitious.
No, one day it happens. Who knows?
Yeah, why
actually
not?
It's not very ambitious. So for example, you have a wafer, so you make the body a slice by a slice, and then you just attach them and you have a human.
Just as easy as a piece of a cake, huh?
Maybe one day. Why not?
Why not? I don't know. Maybe I cannot see that day, but hopefully.
That would be a very scary day.
Yeah, zombies on the street.
That would be a very scary day, but with that, we go to the takeaway. What should our takeaway be?
It's me?
No, I'm asking Houman.
Oh, takeaway for me from TikTok?
For basically our takeaway from this conversation.
Oh, okay. So, yeah. So the most important takeaway is that, we are trying by this technology, for example, organ on the chip that we talked. It was the focus of the discussion. But I also talked about artificial tissues for transplantation, but we didn't go through that during the question. But let's focus on organ on the chip. With these platforms in the future, we have more efficient, safest drugs that we can have, because most of the drugs, as I mentioned, 90%, more than 90% of the drugs fail during this drug development pipeline. And some of them, when they reach even to the market, they have side effects. That is kind of very dangerous. You know, I gave you one example only about the Vioxx that really people lost their life, 20,000 people only in US. So the takeaway is that this organ-on-a-chip model can really help us for discovering new, in a screening and discovering of more efficient and safest drug candidates.
Thank you.
Hopefully in near future.
I appreciate it if you can send that in 1 or 2 sentences to Mohsen in English. So thank you for everyone for coming. Thank you for contributing to this discussion. I guess the next Tech Talk would be in person. Am I right? Mohsen?
I think so, not finalized yet.
Yeah, it would be in person. For in-person Tech Talks, unfortunately, we tried hybrid. It didn't work out, so it would be in Vancouver, in downtown Vancouver, and we will post the video afterwards. But yeah, follow Tech Talk on Facebook, LinkedIn, Instagram, Telegram, and YouTube. And we will post the recording of this session on YouTube and Facebook and Instagram as well.
I'll be happy to fly over for next week in Vancouver.
Please,
we would be happy to have you.
Yeah, we definitely we welcome you.
I've never been there.
You missed out. Vancouver. West Coast is the best coast.
I was in Victoria, but not in Vancouver.
Victoria. Yeah, I like Victoria better. Yeah, I think that's pretty much it for today. Yeah, we're looking forward to see you next week. Oh, by the way, we have a party for those who are in Vancouver. Tech Talk has a party on Saturday, and if you like to join, please message myself or Mohsen. We have our LinkedIn's now, or you can use other means to contact us. I'm sorry, Houman,
If there is only one reason to come to Vancouver, is a Tech Talk party. I have to join.
Most important reason, I would say, but there are other reasons too. Yeah, it's all right. It's an all right, okay city. Yeah, have a good evening or good morning from wherever you join us.
For those who joined us from Taiwan and Iran, I really appreciate, and for those who joined from Vancouver, Waterloo, I think, and Tennessee, I really also appreciate your comment. Old friends from California, Amir, and Mohsen and Amir for invitation. Thank you so much. It was a pleasure meeting you guys. Hopefully next time in person, so we can really see each other. Today I was in a conference. I tried to touch people. Are they real or still like?
It's weird after a long time.
We exist. I can guarantee you that. Yeah, have a good evening or good morning.
Yeah. Take care.
Bye. Take care.
See you. Bye-bye.